Hello friends, welcome back to my YouTube channel Lesson Parasalan. This is a part 3 video. Currently we are revising the important topic for BARC OCES Biosciences examination. Okay. So in human, the inherited autosomal recessive disease, xeroderma pigmentosa, is a result of defective. This disease occurs when there is a defect in this repair of UV damaged DNA. Okay. So if you take the xeroderma pigmentosa, it is due to the defect in the nucleotide excision repair system. Okay, so this nucleotide excision repair system is generally involved in correcting or repairing the damages that was caused by UV light and cross-linking agent. When this UV light or cross-linking agent, when they mutated your DNA, means the nucleotide excision repair system will be taken care. If there is any defect or if there is any mutation occurring in this nucleotide excision repair system, means then the person will be getting xeroderma pigmentosa. Okay, whereas if the DNA is affected due to mutagenic agent like oxygen radical, intercellular reaction, alkylating agent, ionizing radiation, means then your base excision repair system will be correcting all those things. Okay, whereas replication errors are corrected by mismatch repair system and your double strand breaks are corrected by homologous recombination repair system and non-homologous end joining. If there is any defect in homologous recombination system, then these diseases will be arising. This include ataxia, Nijmegen-Breck syndrome, Bloom syndrome, Rothman-Thompson syndrome. Okay, whereas if there is any defect in non-homologous end joining, means then your Werner syndrome and ligase 4 deficiency will be occurring. Okay, so this is an important thing. Repair system is very important regarding to this BARC examination. So the DNA mismatch repair system, if there is any defect in this DNA mismatch repair system, means then your hereditary non-polyps correlated cancers will be arising. Okay, this mismatch repair system is involved in correcting your replication here. Okay, whereas this Bloom syndrome, Fanconi's anemia, hereditary breast cancer will be arising if there is any defect in the repair of double strand by homologous recombination. Okay, so the correct answer for this question is this: xeroderma pigmentosum is an autosomal recessive disorder which is arising as a result of defect in the repair of UV damaged DNA. Next, ETC has a potential to produce highly reactive free radical that can damage the cell. Which of the following is useful to prevent the oxidative damage in cell? Okay, so you should know this reactive oxygen species include a superoxide and hydrogen peroxide. These reactive oxygen species will be generated in cells by either by a mitochondrial source or from any other enzymatic source. Okay, so if this particular reactive oxygen species has not been released or has not been removed from the cell, then it will be causing oxidative damage to a DNA, protein, and membrane lipid. Okay, so this glutathione peroxidase is an enzyme. It is an intercellular cytosolic antioxidant enzyme. So the main role of this particular enzyme is that it will be enzymatically reducing your hydrogen peroxide into water. Thereby, it will be limiting the harmful effects of this particular reactive oxygen species. Whereas if you take this option C and option D, this antimycin A and rotenone are called as inhibitors of electron transport chain. Okay, rotenone is a fish poison and it will be inhibiting complex 1 of electron transport chain, which means it will be preventing the transfer of electron from complex 1 to ubiquinone by blocking the ubiquinone binding site. Okay, so next is antimycin B, which will be inhibiting complex number 3. Okay. So this antimycin will be binding to the QI site of cytochrome C oxidase. So this antimycin will be inhibiting the oxidation of your ubiquinone. Whereas the cyanide and carbon monoxide, azide, hydrogen sulfate, all are coming under poisonous group and they will be inhibiting the electron transport chain by affecting the complex number 4. Okay, so they will be acting on complex number 4 and these poisonous substance will be inhibiting the electron transport chain by binding strongly than oxygen to FeCU center of cytochrome C oxidase. Antimycin A will be binding to cytochrome C reductase, whereas the cyanide and all other poison compound, they will be binding to cytochrome C oxidase. Okay, so it will be preventing the reduction of oxygen, whereas this antimycin A will be inhibiting the oxidation of ubiquinone. Okay, regarding to oligomycin, again this oligomycin is a macrolide antibiotic which is synthesized by the Streptomyces species, and this particular oligomycin, it will be affecting the complex number 4. Okay, so it will be inhibiting F0 subunit of ATP synthesis. Thereby, this oligomycin is involved in preventing the ATP production. Next, which are the following correct hierarchy of gene activity in early Drosophila development? Okay, so this is a correct order. First, your maternal gene will be activated like your BICOR and nanos. Next, followed by GAP genes, maternal gene and GAP gene, parallel gene, segment polarity gene, and homeotic gene. Okay, so this GAP gene, a parallel gene, a segment polarity gene, homeotic gene are called as zygotic gene, whereas this maternal gene will be activating before the process of fertilization. The correct answer is maternal gene, GAP gene, pararule gene and segment polarity gene and homeotic gene. Next, a Nobel Prize in Physiology and Medicine for the discovery that mature cells can be reprogrammed to become pluripotent was awarded to. So it was awarded to John and Yamanaka. Okay, so they were awarded for Nobel Prize for discovering the induced pluripotent stem cells. Whereas for the last academic year, the Nobel Prize in Physiology or Medicine is given to this particular person for discovering the genomes of extinct homins and human evolution. Okay, and regarding the preceding year, like Nobel Prize in Physiology and Medicine for the year 2021 was awarded to David and Adam. For both of this person, they had awarded Nobel Prize for discovering receptors for temperature and touch. Okay, so next, cancer cells have a unique property to exhibit uncontrolled division. This is primarily due to, okay, so cancer cell, they don't have a contact inhibition. Okay, so they don't have contact inhibition. That's a unique property. So these are the other property of cancer cell. They will be having a sustained proliferative signaling. So they keep on growing and they will be evading the growth suppressor. So in a normal cell, there will be many checkpoints. So this cancer cell, they will be simply evading all those kind of suppressing checkpoints. Next, they have a replicative immortality because they have a unique ability to synthesize a telomerase enzyme. Okay, in normal cell, the telomerase amount will short to reduce after each and every cycle. Okay, whereas for cancer cells, they keep on growing without any kind of apoptosis or any kind of cell cycle arrest because they will be synthesizing an enzyme called telomerase. Okay, next they have a property of invasion and metastasis, and they have a unique property of inducing angiogenesis, which means they have a property of forming new blood vessels. Okay, and they have resist cell death. They don't have apoptosis itself. At the same time, they are anchorage independent and they don't have contact inhibition. Next, SSR is said to be a dash type of marker. Markers are classified into two types. Okay, one is called as a dominant marker and another one is called as codominant marker. So regarding to in research side, mainly they will be employing codominant marker because codominant marker they will be clearly distinguished between homozygous dominant like AA, homozygous recessive like small a. At the same time, by using this codominant marker, you can also predict the heterozygous state of a particular allele. And regarding the example of this codominant, sorry, the codominant spelling is correct. The spelling is wrong, friends, so please correct them. Okay, example of codominant marker include RFLP, SEAR, SSR, SMP, STS. These are coming under codominant marker. So that's why this RFLP, SSR are widely used in a research scale. Whereas regarding to dominant marker, so they will not show any kind of allelic pattern, which means the results will not be that much detailed. It will be just showing whether the allele is present or not. Whereas a codominant marker, you can able to easily distinguish between homozygous dominant, homozygous recessive and heterozygous. The example of dominant marker include RAPD, RFLP, EST, ISSR. So its SSR is said to be a codominant marker. Next, the following plant hormone is synthesized from an amino acid precursor. So the correct answer is ethylene and auxin. So if you take ethylene, it is produced from methionine, which is a sulfur containing amino acid. Next, auxin is produced from tryptophan. Okay. Next, abscisic acid, the precursor for abscisic acid is carotenoid, and for cytokine, this cytokine is generally formed by a isopentyl group. Okay. So the cytokine and gibberellin, they are formed from a terpene pathway. Okay. The gibberellin are synthesized from a geranyl pyrophosphate. Okay, so here you can able to see amino acid precursor include your IAA, that is auxin and ethylene, and lipid. Okay, jasmonic acid, the precursor molecule is alpha linolenic acid. Okay, whereas a cytokine, brazinosteroid, abscisic acid, stringonolactone, gibberellin, they are formed from a isoprenoid pathway. Next, which of the following diseases the recombinant vaccine is available? So currently the recombinant vaccine is available of hepatitis B. So these are the types of vaccine, the disease for which the vaccine is employed. If you take live attenuated vaccine, it is employed for measles, mumps, rubella, yellow fever, oral polio, typhoid, rotavirus, BCG and varicella zoster. Whereas killed organisms employed for pertussis, hepatitis A, rabies and so for Japanese encephalitis, both live attenuated and killed organisms are employed. Regarding to toxoid, This vaccine is currently employed for curing diphtheria and tetanus. And regarding the subunit, this include purified protein derivative or polysaccharide or peptide from those kind of organism, or recombinant protein is currently employed for hepatitis B, meningococcal and pneumococcal and typhoid. So for typhoid, both live attenuated is employed, at the same time subunit vaccine is also been employed. Regarding to virus-like particle, this vaccine platform is employed for curing your human papilloma virus. Okay, so outer membrane vesicle vaccine is employed for group B meningococcal, and regarding to protein polypeptide conjugate is employed for Haemophilus influenza as well as typhoid and pneumococcal meningococcal. Regarding to Ebola, they are using viral vector vaccine, and same for SARS-CoV also they are using viral vector vaccine, and for SARS-CoV-2 they are employing nucleic acid vaccine. This include either a segment of DNA or mRNA. So one example is Pfizer, Biotech and Moderna vaccine are based on mRNA based vaccine. Okay, so for regarding to COVID vaccine, these are the list of vaccine that are employed for corona. So Covishield, it is manufactured by Serum Institute of India, and it is a viral vector based vaccine. The viral vector employed is adenovirus. Okay, it is an inactivated adenovirus which holding a segments of coronavirus. So regarding to Covaxin, it is produced from BARC Biotech. Shield Serum. Covaxin is a vaccine produced from BARC Biotech, and it is the whole virion inactivated vaccine. Sputnik, it is produced from Russia, and in India, Dr. Reddy's laboratory is licensed for manufacturing the Sputnik, and it is adenovirus vector vaccine. Regarding to Corbevax, which is manufactured by Biological E Limited, it is called as protein subunit vaccine. So this the vaccine is having a receptor binding domain of SARS-CoV-2. The protein subunit is nothing but is a receptor binding domain of a SARS-CoV virus. Regarding to Zygo-VD, which is produced by Zydus Cadila, it is a recombinant DNA novel coronavirus. So please try to know about the list of vaccine currently employed for the coronavirus. Both you should at least try to know the list of vaccine currently manufactured in India. So in India, currently they are employing a nasal route based vaccine. So you should know about all those things.